Welcome, everyone. Thank you all for taking the time today. We're going to start by introducing the growing point. The start of this webinar series and the goal of the Alberta Wheat Commission and the growing point, which is the Alberta Wheat Commission and Alberta Barley Commission's agronomy extension network, is to really provide hands-on agronomic information for producers in the industry to utilize. My name is Jeremy Boychin. I'm the Agronomy Research Extension Specialist with Alberta Wheat and Barley Commissions, and I'm going to facilitate the session today. And I'd like to introduce Dr. Reem Aboukhaddour, who is a research scientist and cereal pathologist at Alberta and Agri-Food Canada. And she's going to talk to us today about stripe rust, some considerations that are important as we head into this next season, and management practices that everyone should know about moving into this season. So thank you for joining us today. Before we get too far into this yet, I just want to give a little bit of background, and then I'll let Reem jump in. So again, as I mentioned, the Alberta Wheat and Alberta Barley Commissions want to take a very hands-on approach to agronomy learning, and the development of the growing point was part of this goal, and the development of this webinar series in a time where we were limited in being able to meet together in the field or in any type of meeting. So we wanted to find a way to bring researchers and bring information to producers and to agronomists and to the industry, and still have it to be timely and relevant. So this was our way of doing this as a webinar series, and getting experts like Reem to chat about what they do research on and why that's relevant to producers. So a few housekeeping things, and I mentioned it as people were rolling in. There's a few different ways that you can interact with Reem and I. They are at the bottom of your screen there. You can use the Q&A button. So if you click that, you can type in a question and it'll come up to me, and I'll be able to ask that question to Reem. And Reem, maybe once you get going, you can let them know whether you want to have questions throughout or save them to the end. And even if you do save them to the end, I encourage people to ask questions throughout the chat and the Q&A, and I'll kind of log those and then ask them at the end. You can also raise your hand, and that'll give me the opportunity at the end of the session for me to open up your mic. So any of the attendees can click that button. I'll open up the attendee's mic, and then they can ask the question verbally. Or you can use the chat function, which a lot of you are already engaging in. So it's great to see that. And again, I highly recommend anyone who's already part of this conversation right now, jump into the chat, change your the to button to all panelists and attendees, introduce yourself, let us know where you're from and what you do. And it's always fun seeing people engage and have conversations in the chat as the presentation is coming along. Those are all the main things. So I will let Reem take over now. Thank you again, Reem. I'm excited to see what you have to say.
Thank you, Jeremy. Thank you, everyone, for coming. I am very happy that I also got the chance to share some of the work we do with you. So I'm going to share the screen. All right. Do you see that screen?
Perfect, Reem.
Good. All right. Good morning, everyone. So I'm going to talk today about stripe rust, virulence, and some management consideration. And the outline of my talk, okay. Oh, here we go. So I will be giving a bit of introduction about stripe rust, the disease and the pathogen, and then I'll talk about why it is so damaging, and then I'll touch a bit about the worldwide situation and compare it to Canada. Then I'll go through some aspects of the work we do in our lab in terms of surveillance, the disease nursery, and the risk characterization, concluding with some of the management considerations. So for those of you who are not familiar with the stripe rust of wheat, also is often known in Australia as yellow rust of wheat. It's a fungal disease caused by a pathogen. The pathogen name is Boxinia striformis forma specialis tritici, and I'll be referring to it as BST today. So this is one of the most destructive foliar diseases around the world if we speak as of today. And the pathogen can infect any green part of the host, or like here we are talking wheat. And the infection, really the symptoms appear with this. You see it tiny orange to yellow colored pustule, or we call it urea, the fungal structure. And it infects the host plant from any stage, really from the seedling until maturity. So when it is on the seedling, those yellow structures are not arranged necessarily in the stripe as it does on the adult plant, and that's why the name stripe rust came from. With the heavy infection, you can't see the stripe anymore because the whole leaf get covered with the spores. Now it's not a seed-borne. The spores microscopically are very small and they light and they travel thousands of kilometers, yet still remain viable to cause infection in totally different geographical area. So, but with heavy infection, you could even see the spores accumulate on the top of the grain between the glands. That's in terms of symptoms. If we were to talk about in terms of damage and in relation specifically to other diseases in Canada, so this image here, it shows you the most damaging, the top damaging fungal diseases in Canada through the time from the start of wheat cultivation until now. Now stripe rust is considered one of the most recent damaging disease since the 2000. That doesn't mean it wasn't occurring before. It was, but I'll explain later why it's became more damaging since 2000 in Canada. Now, in terms of geography, where does it occur? It's kind of restricted in Alberta and in BC most often, but recently it has been also showing more to the eastern prairie. And this is this map, it shows you where the area of wheat, like this color area, and there are beside the stripe rust on wheat, if you know, there is the stem and the leaf rust. Those are very important diseases, and you could see those ones are known for over 100 of years. Now, those two types of rust do occur mainly in Manitoba and southeast Saskatchewan. This like area here is called the rust area, we know by that way, and they come through really south of, through the Great Plain, Texas up to Dakota, and then to that rust area, and they are usually here. But with the stripe rust, you have it in, as I say, in BC and Alberta, and that's actually exactly why we have the disease nursery for stripe rust in Lethbridge, and for the other two, it is in Winnipeg. So just a bit of adaptation for the different diseases. This one, it prefer more that climate of cool and moist spring for infection. Those ones more a bit of higher temperature. Now, if I were to look at stripe rust compared to the other two rusts, stem rust and leaf rust, in terms of the number of years with moderate and severe damage over the past 100 years of wheat cultivation in Canada, again, you could see I didn't put stripe rust because we don't have that data. It's not known to as damaging before the 2000. But you could see with the stem rust, you can see like the damage has been quite high, both for stem and leaf rust, and very well known until like with the stem rust, it's ended by the mid 50 due to the successful genetic resistance. With the leaf rust, I would say since the 90s, there are a few outbreaks, but it's also as well managed with combination of genetic resistance and fungicide. Now, if I were to talk about stripe rust from a global perspective, so this image here shows you before 2000, the situation with the stripe rust severity and intensity around the world. the more intense color, the more severe the disease was. But what you could see, it was more endemic. So it was more restricted to region with high elevation, cool and moist. And it was known to occur, for example, in high elevation area of the Pacific Northwest, or some high elevation area in South America, Chile, Argentina, and Brazil. And then in Atlantic Europe, some high elevation in East Africa. China was well known for the damage, and also Nepal area. In Australia, since the 1979, there was inclusion of frost in that continent. So what happened after 2000, there was a new races that emerged to adapt to cause infection at higher temperature, which is not typical of the pathogen. What happened with that, the disease was able to expand its geographic boundary and became problem, more intense issue, and became of problem really wherever you grow wheat around the world. So that's a little bit about why it's become more of a damaging. In terms of cultivars around the wheat, 88% of the wheat cultivars are really susceptible to it. The annual loss is estimated by $1 billion. So where this disease originates, it's believed that stripe rust originated thousands of years somewhere in the Himalaya region. And from there, it is spread all over Asia. And then about 100 years ago, those strains from Europe theoretically could come to the USA and to the America, and then to Australia, to Western Australia. What happened around 2000, the East African or Middle Eastern isolates that are represented here in red, those are the ones that are adapted to heat, and those ones spread all over the different region and really replaced the older blue lineages. And they spread also to USA, then to Australia, and also to Canada. So if I were to say who is working on stripe around the world, there is a whole lot of people working around it, but there are a few labs that take care of the doing race characterization and communicating about the virulence around the world. Basically, there is something we know as a researcher, we call it the STOPS collection. STOPS is the last name of a scientist who was in the Netherlands. So they did a lot of work in Atlantic Europe since a little bit before the Second World War. And they have more of international a collection of more than 5,000 isolates. When STOP retired in the 1990s, that collection went to Denmark. And they did a whole lot of work, and they expanded. There is, I think they have over 6,000 of these different isolates. And the reason why I'm talking about that, because they provided a lot of the images about the origin of the pathogen and etc. by doing a lot of the research and the kind of fingerprinting of the disease. You have the Australian, they have a lot of work on the rust. They provide a lot of essential material for research. And you have the North American in Washington as well. The other location where they do more of a field kind of like applied looking for adult plant resistance is where they do in Semet, in Turkey, and in Morocco, Icarda and Semet. They do a lot of that work. We do as well since 2000 like in Canada. So a lot of the programs here I'm gonna mention, if you are interested with more elaborated information on rust, you could refer to the Global Rust Initiative, where they have a yearly meeting for really researcher working on all cereal rust, including the stripe rust. Washington State University, they have very important info, especially for people in North America. They might find it very useful resource. So the aim is to develop genetic resistance, and for us you have two types of genetic resistance. We call it the adult stage or the field resistance basically, and the old stage, which is expressed from the seedling stage until the plant is adult. This one is not expressed at the seedling stage, so it doesn't give protection at early stages. However, it's very effective because it's durable, not easily broken, and it protects your adult plant. Now, it doesn't give 100% resistance, so it's not like immune or fail. So the plant can endure a bit of infection, but still can survive it, and really with the minimum influence on the yield. With the olive stage, it gives you pretty much very good resistance, like fail of infection. However, it's not durable. The pathogen evolves virulence against this quite often, and so it is the challenge of the olive stage resistance, that its lack of durability, but it does protect your crop from early on. In my lab, we try to look at both aspects actually. So for the adult or the field resistance, what we do, we do the stripe disease nurseries, which unfortunately this year I have to show it on a slide. So we have this large plots where we test large number of genotypes. Usually over 25,000 wheat genotype are tested. This one are shared from different breeding programs. They give it to us so we can test it under a high pressure of disease. That means you apply the pathogen and you ensure there is a high pressure disease, that you can tell the difference between your susceptible and resistance. Many people are involved in this work, many technicians and students. There are many factors to take in consideration when you want to establish your disease nursery successfully, from the agronomic practice to the in-column viability, and that it represents the dominant virulence in that year being homogenized. At the end, you want to have the maximum expression of the disease, so you tell resistant line from susceptible when they are side by side. The process is never ending because the pathogen keep changing in virulence. So every year we have to screen, because whatever resistance you could have, it could be defeated, and you need to know that. The other type of things we also do is to go survey, and I think this is very important. I do it for Southern Alberta, and then I collaborate with numerous colleagues in Central Alberta, Mike Harding, and then in the North. And what we do, we try to go in different fields through the growing season, and we take a data based on the severity and incidence. We classify those different fields into clean, like having no infection, or having light or trace infection, which usually is less than 5%, moderate 5 to 10, severe anything over 10. And then we take that data year after year. I started in 2006. I could do it for the 4 years, and it can give you information about how important this disease in certain region per year. So the accumulation of this data long term will provide really good solid information. As you can see, 2016, if you remember, was very bad year. 17 not that much, until like last year was as bad as the 2016. What we do as well during this survey, we try to collect samples of infected plants from each of these different locations. We take that and we try to answer how that virulence changed in Canada. So we mainly relied a lot on the USA people to provide that data to us. There are sporadic research, however, some in the University of Saskatchewan do provide that as well, and the provincial people in Alberta. However, I wanted to look more of over the last 30 years, something strength we have in AFC, that we have a historical collection. So my aim was to answer, how did the pathogen really change in Canada over the time, and was it similar to the USA scenario? And given the fact we have also some isolate from the East, are there is any variation between the East and West in terms of the virulence? So basically, we explore, this was a, so the previous slide was already published work. We explore 140 different isolate from those different time and from the different provinces. So basically you collect the pathogen, you increase it on the plant, and then you inoculate different wheat genotype. Each wheat genotype supposed to carry one of the resistance gene, we call it YR gene. And those genes, kind of standard, they are used by different research groups around the world. It's kind of language we communicate with it. I'll explain it in the next slide. So what you do, you inculate your different lines, and you kind of like rate them on a scale from 0 to 9 for infection, 0 being very healthy, 9 being the most extreme susceptibility. And we color code them: green for the more resistance, the more you go, yellow and orange is more susceptible. So let's say we use these different lines that have different YR genes, yellow rust resistance genes, as expressed in this horizontal here. And those are the different isolates. They are color coded by the time. So if you look at the color, you could realize before 2000, pretty much everything, almost like the majority, were in the green, so not defeated. But since 2000, you could realize more of the orange and yellow. And then since 2010, it's also again more of that. And that's just giving you the visual representation of how the stripe rust pathogen population in Canada shifted to more virulence. Particular since 2009, those races that we talk about that they came from the East Africa or Middle East, they are characterized by defeating those two genes, YR8 and YR9, and that's pretty much what we see. 2010, we got YR10, which is in winter wheat, also partially defeated, I would say. So this is this is type of information. When you have it, you can have for each race or each isolate what we call it the virulence formula. So you could say, well, I have this and that defeated / this and that not defeated. And when you communicate that with the different researchers around the world, you can trace if there is race inclusion or not, because most of them, they use kind of the same standard set of genes to characterize that. So in other words, if we also were to apply it for the breeding side, you look at the different genes and you can see in time. So the first horizontal line for the isolate before 2000, and the yellow represent the percentage of the of isolate that defeat this particular the genes are here represented. So you could see that from 2000 to after 2000, you like before 2000 you got this gene defeated, then after 2000 you add to them those color coded in red extra 5 ones, and then after that you add another 4 ones. So you have quite out from the 18 gene, quite a lot of them are defeated right now. When we look at the different races, we did realize that per time there was different dominant races. Since 2010, the most dominant races in Canada are really the same races as in USA. However, we have low frequency of unique races in Canada, meaning that the pathogen can evolve different at local level. And that's also important to know, because likely that is under selection pressure from whatever different cultivars we grow here as well. Now, if we look at the different isolate races per region, we realize the one that in Alberta were very, in terms of virulence, cluster very closely to the one that coming from California and Washington, so the Pacific Northwest, so they are more related to that region. The one that we have from Ontario and Manitoba, they were more close to the Dakota and Nebraska, so they were more close to the Great Plain, and that basically pretty much confirmed the hypothesis that now stripe rust come to Canada on two type of wind trajectory. You have the one that coming from the Pacific Northwest to South Alberta and BC, and you have the one that come with the Boxinia, typical Boxinia pathway with the stem and leaf rust through that to the eastern prairie, and after that to eastern Canada. So in conclusion, we know that stripe rust virulence has changed in Canada and increased since 2000 and again around 2010. And most of the dominant races really much similar to the USA with a few unique ones. The race structure per province vary because of the different races arriving on different wind trajectories. And that's pretty much the story. Now, this collection that we have that represent the last 30 years is really can be considered as a research resource for fingerprinting the rust and enhancing more advanced diagnostic tool based on molecular marker or some work that can be done to detect the pathogen in a faster way than just the bioassay. These races, having these specific races is a powerful tool where we initiated a lot of collaborative projects to look at the seedling and the adult stage specific resistance and mapping of other resistance sources. And at the end, it's important as a coded language to communicate new virulence inclusion with different researchers around the world. In terms of management consideration, I would say probably if you have Kelly Turkington and Mike Harding, they are very strong in this area, but pretty much genetic resistance is very important tool. Research has shown that when you plant a range of cultivars, whether it is very susceptible to resistance without fungicide application, you would see variation in the potential yield losses could be up to 40 or more. You could see like 80% damage when you have a very susceptible line versus, let's say, 30 when you have moderate resistance to moderate susceptibility, or if you have moderate resistance, you have 0. This is a research done in Australia. Other management practices is really the disease monitoring, and I think Kelly probably would have talked about that. We have the Prairie Crop Disease Monitoring Network where they release weekly or bi-weekly information. It's predicting the risk of rust based on the information we get. So monitoring the disease, we provide data, or USA provide data, and then the wind trajectory and the weather parameter, and they release that. They have a Twitter link for that. I think it's very useful for wheat grower to look at that very carefully at the growing season. Really not much other thing is more of avoid early planting of winter wheat. If you are in an area where stripe rust is very risky, you don't want that green bridge, because winter wheat can be infected first and then you have a chance to build up inculum and transfer it to your spring wheat. So reduce volunteer plant and grasses, because that also, the stripe rust can overwinter in those hosts. With any fungal disease, they like a lot of moisture and green tissue, so no need for excessive irrigation and fertilization. And at the end, you want to protect the flag leaf, because that's the flag leaf and the head give you up to 60% of your yield. So that's pretty much all of the management reconsideration I do have. And I want to acknowledge my group who help a lot in this work. Ryan Gorley, who is the lead technician on a stripe rust. Eric, who is another very good technician in our research station, helped a lot. And other member of my group, postdoc and, biologists and students. And a lot of collaborators and colleagues throughout Western Canada. Particularly the stripe rust, the cereal rust pathologist in Morden and in USA, Dr. Chen, and a lot of people from provincial and other places. And for sure, the funding from Agriculture and Agri-Food Canada, the consortium funding Alberta Wheat Commission, Saskatchewan Wheat, and Agriculture, Forestry, and Western Grain Foundation. That's all. Thank you very much.
Thank you, Reem. That was wonderful. I want to open up the floor now to questions. If anyone has any questions, feel free to either share them in the chat or in the Q&A. I know I have a couple questions that have popped up, and I know we chatted about stripe rust and it felt wheat kind of concentrated. Could you maybe hit on what are the points between barley and wheat? Are we dealing with the same issue here? Like, or should we be considerate about barley in this conversation as well?
Well, barley is caused by a different forma specialis. So the fungus has different forma specialis, or different form. One of them attack wheat, that we call it forma specialis tritici, and the one that attack barley, we call it horridae. In terms of barley, I would say is not known as damaging as it is to wheat for some reason. However, like the same consideration, sometimes you get some of the form that attack barley could attack wheat and vice versa. So it's not like there is a definite yes or no. But usually the forms that are on wheat do attack mainly wheat, and the form that on barley do attack mainly barley, but sometimes you get that cross in terms of management, in terms of dealing with the pathogen is pretty much, I would say, the same, except that what I know that the disease in barley, stripe rust on barley, is not as important as on wheat. I know that my colleague in Lacombe, the provincial side, especially Kekwan, he is now in Olds. They did a quite of interesting work on barley as well. So that's.
It's just we think about scouting fields for stripe rust, and where should we be? There's a lot of fields to be checking and a lot of things to be looking for. Should we also be monitoring those barley fields and keeping our eyes open for stripe rust there as well?
I do, actually, and yeah, you are right. We should, and we do.
Okay, perfect. So I had a couple questions here in the chat. The first one is from Samia Fatima. What types of testing is available for growers? And I'll add on to that. What benefit could maybe not just the research side, but also the producers get from that testing if they wanted to do testing?
You mean testing for what testing? I'm sorry.
Well, maybe
I don't see.
that was the whole of the question. I guess I'm assuming.
Oh, okay. What type of testing is available for growers? Well, you know what? It's just from perspective of the growers, they want to know if there is a stripe rust first or not. They want to know if do I need to spray my field for stripe rust or not? Is my cultivar gonna be resistance or not? That's why we have the disease nursery, and that's why the breeders always keep to screen their new release cultivar for it, because at the end, what is provided out from that is seed guide, where you have the different cultivars with their baggage of resistance to the different diseases. And then with that, they can decide if they are in an area where a stripe rust consider of high importance, like I would say in Alberta, in South Alberta, then they can definitely pick of more of moderate and moderate resistance, or at least intermediate, and grow it than having the susceptible. And that, as you can see, can give you quite more of assurance. In terms of growers want to test for, I think the races is really at the end, it's just the step before it get applied. It's more for, I don't think the growers are really gonna look of what is the race. That's not their concern. That's my concern and the breeder concern. So we are aware if there is a different form of virulence. So we try to see what went wrong, which resistance gene defeated, and how we can build a new resistance or search for a different source. So that's the way I see it.
Yeah, I would imagine, if someone did find stripe rust in their field, it would be less of a question of, should I get this tested for stripe rust, or what race it might be. It should be more of a question of, which variety do I have? What resistance level am I dealing with? Am I in an environment where this is going to continue to become an issue? And how quickly do I need to address this, rather than being concerned about what race it is? Because we do know once it gets to that 5% leaf coverage range,
yeah.
Significant yield impacts can happen. So rather than being concerned about race, it should be, how quickly do I need to make this decision based on the variety I have?
Yeah, but we are I'm always interested in getting samples of stripe rust. So whenever I get a sample and the producer have sent, I mark it where I get it from, and we eventually the data become available what virulence it has. So that is also important.
Perfect. We got a couple more questions here I'd like to get through before we have to leave, but a question here from Sandeep. Please tell me about the preservation and revival of the Uridino spores of stripe rust. And yeah, I think I said that right.
Okay. Yeah, so you can collect the spores. There is a certain tool or a machine you can vacuum them. And what you need in terms of preservation, really, you want to first to dry them pretty much, and then to preserve them for long term, you have to put them in liquid nitrogen. Some people do in some, you can put them in -80. They would not survive many years. I mean, the 30 years old isolate, they were in a liquid nitrogen, and that's pretty much a standard with any type of rust. But they have to be pretty much dry spore, and before you use them, you need to give them back that moisture to revive them again. It's just the biology of the fungus.
Perfect. I will jump over to Lindsay's question here. Is there a scouting guide available to help determine the difference between all of the rusts?
I pretty sure assume there should be. It never come to my mind. I'm sorry, I should have provided a bit maybe slide at the beginning about what is difference. They are very easy to tell apart. Like stem rust is actually mainly on the stem, only under heavy infection you get it on the leaf. It look more of a dark color, more like brown dark color, and is pustular, kind of like bigger. With the leaf rust, it's more of the brown, also like brick kind of color, and it's all over the leaf. And it's pustular also a little bit bigger than the stripe rust. Stripe rust is very much distinctive. It's almost like that yellow, and it's in stripes. So it's very distinctive to tell those three different rust apart. I find it that way. Jeremy would know about that. You would know about you did the book about that, right? You provided.
We have and I was actually just looking to find the link for that. There is we did a spotlight on diseases a couple of years ago that I believe had all three end images in there, and we are looking to update that information that's in there as well. And I was actually just looking on the Prairie Crop Disease Monitoring Network. And I don't think Kelly has all of those in there. I mean, you're going to find, if you look up anything in Alberta or wheat or in Saskatchewan, you're going to find a lot of visuals and information on stripe rust. But yeah, I would say, leaf rust and stem rust, because they're not as common of an issue recently, you're not going to find as much information on them. But I couldn't find that link for ours, but maybe I'll try and post it up after.
And if you want to contact me, I can provide you with tons of information on both. I could type my email in the chat and just contact me.
Yeah, I think that'd be great. And then we can, we're at 11:41 now, so I think we'll end here. And while you're typing that in there, Reem, again, thank you so much. A reminder to everyone who's still on here, this video was recorded, so it'll be up on the Alberta Wheat and Alberta Barley Commission's YouTube channel. And we do have our next In the Field webinar on June 2nd, and we're going to have Maria Constanza Flietes, and she's out of the University of Saskatchewan, and she's going to be talking about bacterial leaf streak scouting and management. So that's going to be a really interesting one, because it's a kind of disease that's kind of sneaking up a little bit and causing some issues here and there. So if you want to share any of the content you learned today on Twitter, you can find me at Boychin Jeremy, and then at Alberta Wheat or Alberta Barley, and you can hashtag them as well. So again, thank you, Reem, and thank you for everyone joining today. It was a very informative session.
Thank you very much. Thank you, everyone. There was one question you missed on the queue.
Did I?
Somebody.
Oh.
Yeah. And I didn't.
Okay. We could tackle that. If you have an idea about the role of wrong practices in disease distribution. I'm not sure exactly what that means. If you have an idea about the role of wrong practices in disease distribution. So what I take from that is what practices might people be doing that may encourage disease spread? It's kind of a very good question.
Yeah. I would say it's more of concern, let's say, if you're talking about club root, where you have transmitted through your boots and stuff like that. With rust, you really, the only wrong practice is have your susceptible cultivar and then wait for the spore to come by air and rain shower on your crop. I guess that's it, or excessive fertilization or extra application of fungicide when it's not needed. I hope that is the question, but I try to answer my best, I guess.
No, that's perfect, Reem. I think scouting and variety selection are going to be so important in this battle. So, okay, well, thank you again, and I look forward to seeing you all on the 2nd for our next session.
Okay. Thank you, everyone. Bye.